Hi, I'm Wei Chong Luo, a fourth-year PhD student at Dr. Shilin Liu's lab. The reasons why you are watching this video are not only because you might be interested in my research, but also because of COVID-19. This unprecedented pandemic has ravaged the whole world and causing more than 2 million of deaths so far, and has forced most of the events to go from in-person to online, like this health fair. Thanks to the advance of the biopharmaceutical technology, the scientists were able to develop vaccines that can prevent this disease from spreading within less than a year. The vaccines that were first being approved for emergency use are the ones from Pfizer and Moderna. Both of them have shown to be highly effective, and one thing they share in common is that both of them are mRNA vaccines. This new type of vaccines makes use of lipid nanoparticles to encapsulate mRNA, and the lipid nanoparticles can protect mRNA from being degraded in human body and then be successfully delivered into immune cells. And then they can later be used to make a piece of the spike protein that is unique to the SARS-CoV-2 virus. It sounds wonderful, right? Unfortunately, both mRNA and lipid nanoparticles are not stable in water. mRNA will undergo hydrolysis, and nanoparticles can gradually form large aggregates in water. So the vaccines require an extremely cold storage temperature to freeze the water for maintaining its efficacy. And this leads to a great logistical challenge because the cold chain is expensive, making the products unaffordable for poorer countries and consumers. And the company also needs to spend more money on the storage. So one of the approaches to improve the stability of pharmaceutical products is freeze drying, also known as lyophilization. So by removing the water from the nanosuspension through freeze drying, the chances of particle aggregation and chemical degradation can be reduced. So the shelf life can be extended. So the figure on the left shows a simple outline of a freeze dryer. So we first load our products on the shelf and then we close the chamber door. And then for the first step, the freezing step, we reduce the shelf temperature to minus 40 degrees C. So the water turns into ice and then the rest becomes a cryoconcentrate. And after that, during the primary drying stage, the vacuum pump starts working. A low chamber pressure is maintained to sublime the ice, and while the shelf temperature is gradually increasing. And for the last step, the secondary drying stage, the shelf temperature is further increased to 30 degrees C or even higher to desorb the unfrozen water from the system. And at the end, we'll be able to collect the freeze-dried products like these shown in the picture. But freeze-drying nanoparticles is not an easy job. In fact, the biggest challenge we face is that as water turns into ice, nanoparticles becomes more concentrated and particle aggregation can occur. And they may even remain after being reconstituted back to the liquid form. So to solve this problem, scientists have found that sugars like sucrose or trehalose can prevent proteins or nanoparticles from aggregation during freeze drying. But which one works better for solid lipid nanoparticles, and how much should we add? In addition, it has also been shown that freezing conditions can also impact particle size during freeze drying. So how fast should we freeze the lipid nanoparticles? And the water in all vials do not freeze at the same time and temperature. So do we need to control it to make them uniform? So these are the questions that remains to be answered, and this is what I have been studying. So from our results, we found that sucrose works better than trehalose for solid nanoparticles. As you can see from this figure showing the changes in the particle size distribution of solid nanoparticles after freeze drying. The left axis stands for the mean particle size in nanometers, and then the right axis stands for the polydispersity index, abbreviated as PDI. The higher the PDI, the more heterogeneous the particles are, so which is a bad thing. And the BF stands for before freeze drying, AF stands for after freeze drying. So before freeze drying, the nanoparticles were around 100 nanometers and the PDI was around zero point 0.1. And without the addition of sugar, the particles could increase to 500 nanometers in size and the PDI around zero point 0.7 after freeze drying. But with the addition of trehalose at a ratio of 1 to 10, the particle size were improved. And with sucrose at the same ratio, the effect was even better. So now we know that sucrose works better for solving nanoparticles. Then we added sucrose to our formulation, and then we freeze-dried them by using uncontrolled freezing and controlled freezing. And then we monitor the particle size distribution for 6 months after freeze-drying and compare that with the liquid form stored at room temperature and 4 degrees C. So the figure on the left shows the changes in the mean particle size for 6 months. So this is the liquid form stored at room temperature. This is the liquid form stored at 4 degrees C, and this is the uncontrolled freeze-dried form. This is the controlled freeze-dried form. So as you can see, at room temperature, the freeze-dried forms are better, more stable than the liquid form, and the controlled freezing showed better results than the uncontrolled freezing. And we also found a similar trend in the change in the PDI values. So we hope that this research can benefit both academia and the industry to have a better understanding about how freeze-drying process can impact the stability of nanoparticles. So that in the future, hopefully there will be more freeze-dried nanoparticle products available to patients all around the world. Thanks for your listening.